Give me a warm round of applause. Welcome you to Professor. All right, thanks, Ed. That was great. So actually, I'm affiliated both with physics and applied physiology, so there'll be some of both of those things in this talk tonight. And what I wanted to do first is get a sense for how many people are from engineering in the audience. Do we have anyone in engineering? Oh, good, quite a few. Excellent. And in the College of Sciences? Yeah. Yes, excellent. Good, almost half and half. And anyone outside of both institutions? Good. Because there are some that, there's some elements of design that actually work really well with animation and things that we'll touch on briefly. But what I want to talk about today is this idea that if you've been paying attention to some of the new technologies that have been emerging, there's this idea of bio-inspiration, the idea that there's things in the natural world that we can look at, learn about, and then adapt to human innovation and technology. And what I want to talk today about is why I, a scientist, may actually care about that, since I'm not actually an engineer, and what the coupling between science and engineering can do for that kind of work. And so I want to start by talking about that it's a really exciting time because we've had quite a number of innovations that have arisen from nature. And so one of the more recent examples is this boxfish here up in the upper left has a very low drag and it stabilizes itself with these keel-like edges along the side of its body. And Mercedes-Benz recently introduced a bionic concept car that had sort of a similar design, and it can get up to about 70 miles per hour, and that's not with a hybrid engine or anything. And one of the classic examples of bio-inspired design is Velcro. So when George de Mestral was walking around in the Swiss Alps in the 40s, he discovered these burrs on his pants and on his dog's coat. And he looked at them carefully under a microscope, and he realized that they were interlocking, but that they could come off if you pulled on them very hard, which is exactly what led to Velcro. In the 80s, there was this discovery of this paint lotusan, or this invention of it, which was inspired by a lotus leaf, which has this very hydrophobic surface, so water droplets beat up on it and will actually pull stains off of the surface. And it's very robust. And so it covers the surface of the lotus leaf and inspired this paint. Maybe we can go for a little larger. See, I didn't turn off my phone. A good reminder to turn off your phone. Yeah, we'll need that later, so there is a reason it's there. So we can go to whales and windmills. If you look at the scalloped edges here, when my colleague Frank Fish, yes, his name's Frank Fish and he studies whales, when he was looking at these guys, he looked at a model in an aquarium one day, and he saw this sculpture of a humpback whale and saw these bumps on the front of the flippers, and was like, no, that can't be right. The sculptor must have gotten it wrong. Those should be on the back side because, you know, everything we've learned about fluid dynamics is you should have a nice smooth surface up there for fluid flow. And it turns out, no, they are actually on the front of the fins. And when he studied them, he realized that they actually help the water, they channel the water down the fin and keep it bound to the surface so that you get less turbulence. And so he spun off a company and started developing in Canada these new turbines that have these same tubercles on them and reduces noise and increases the efficiency. And a similar thing happens when we look at shark skin. Shark skin is actually not smooth, even though it feels like it if you run your hand in the opposite direction if you've ever touched a shark. It actually has these denticles on it, which have these sort of, I don't know, this always looks like some kind of spaceship from Star Trek to me. These channels here channel the water down. You can see they're aligned, and the water flows down through that surface, and it actually keeps the water, again, bound to the skin of the shark. And that led to these new innovative swimsuits that people used in the Olympics. And you might have remembered this from the Olympics a couple of times ago, where people were increasing their swimming speed and were breaking all those world records in part due to having these new suits. So this is great. It's wonderful. You know, we can just go out into the biological world and we can do what Da Vinci said. We can just look out there and we can say, well, you know, human ingenuity, it's great, but you're never going to make anything that's like nature. You know, we'll never make various inventions. We'll never devise anything more beautiful, nor more simple, nor more to the purpose than nature does, because in her innovations, nothing is wanting and nothing is superfluous. Yeah? Well, here's a list of the 20 greatest engineering achievements of the 20th century. How many of those are biologically inspired? Maybe flight. That was actually bicycle inspired. And airplanes don't really fly anything like the way that birds do. We'll talk about that a little later. There's a little bit of ideas in here that might have come from maybe somebody looking at nature, but there wasn't really anything where people were trying to understand particularly living systems and adapt it to new technologies. And that's because human engineering and natural technologies are really different, particularly in the 20th century. In the 20th century, there was a big focus on making large technologies, large constructs, large robots, large networks, large materials, whereas natural technologies typically are small. So the average animal is actually only 5 millimeters big. This is sort of true. We can see that also in the design of systems where human engineering typically uses flat right angles and natural surfaces are more curved, in part because they're interacting with very inhomogeneous environments like this muddy, sandy material. And as a result of that, human technologies tend to be stiff. Again, like this airplane, you can get a little flexion in the wings, although a lot of people don't like to look at that when they're flying. And but it's nothing like the wings of this hawk moth here is hovering in front of a flower. So this is slowed down 30-fold. And you can see how much bending and twisting there is in the wings as it flies along. And humans really like rolling devices. I think this is a Camaro. I'm not much of a car person. So, but I do know what this is. This is an amblypygid. And if you've never seen a whip spider before, this is actually a spider. So it has 8 legs. And natural technologies tend to use a lot of appendages. And these guys are particularly interesting because while they have 8 legs, they run like an insect. They run on 6 legs. And that's because the front 2 legs have become these foot-long antennae-like structures that it uses to probe its environment. And it will sneak up on prey like a little cockroach, and it will tap it, and it will tap it. And then it will use its 8-inch wide mandibles to lunge out and grab it. And so this guy right here is about the size of my hand. The last difference is that human technology really has, this is the Honda Asimo robot, really cool robot, but it tends to rely on few actuators and few sensors per joint. Whereas, particularly if we look at ourselves, we have many muscles and many sensors per joint. Why do we have 4 muscles all moving our heel in the same way? You know, what is it about these natural systems that has this level of complexity to it? And so perhaps what we can, but perhaps what's happening is as the 21st century is rolling around, our technologies are changing. Things are becoming soft, things are becoming flexible, things are becoming multifunctional, reconfigurable. You've probably heard all of these terms being involved in the tech community a little bit here. And so perhaps what Vogel said in 1998, which is that as human technologies take on more of the characteristics of nature, nature can become a more useful teacher. And so that's what I really want to explore today, is I want to say, how do we learn from nature? And what is it that we can sort of do as a program, particularly as a scientists and engineers, to maybe see how we can transform what technology will be like in the 21st century and what science will be like as well. And so the first thing I want to say is I want to go back to that quote from Da Vinci and query, why is that not the case? This is something I generally like to ask, which is whenever somebody posits something like, oh, natural technologies are wonderful, or you can do so much with natural technologies, I usually like to ask, well, what can you not do with them? Or everybody wants to know how the brain works. I generally like to know how the brain doesn't work because it's incredibly flexible. It can do lots of different things. All right, so what are the things about biological systems that we have to keep in mind when we're doing biological inspiration? Well, the first thing comes in that biological form and function is a tricky business, right? So if you take a look at this bird and you look at these beautiful long plumose feathers that are coming off the tail here, you could go through and you could say, well, there's probably a lot of aerodynamic functions that those feathers could have, right? They could be increasing they could be decreasing wing loading. They could be increasing the amount of surface over which fluid can flow so as to increase lift. They could have special structures on there that decrease drag. But actually, no. They're there because they're sexy. This is a bird of paradise, and it actually suffers severe aerodynamic consequences for having those really long feathers. And in fact, it's sort of been shown that a lot of biological systems will actually elaborate these really big structures like a peacock's tail that probably don't have a function in terms of locomotion, but are actually evidence of just how fit that animal is, because look at how much energy I can dump into making long, big tails so as to make myself more sexy for my mate. So we have to be a little careful when we say, well, what is the function of something? And that's also true when we consider that organisms must grow. Generally, a lot of robots don't sort of start off in a nymph form or a worm form and then, or a centipede form and then become a moth, for example. This is something pretty unique to nature. And in fact, you can see really odd things that happen because of it. So this is an adult locust here. And when you look at the adult no longer flies, and it's lost its flight muscles in order to make room for its legs, but it still has the flight muscle motor neurons. So it still has the neurons that would go to those flight muscles, but they don't actually go anywhere. They just sort of hang out there in the adult. And that's because the biological system is facing a developmental constraint. When it's developed, it hasn't been able to turn off those genes because they also control other genes in the animal that are critical to its function. And so biological systems are not necessarily free of what we might think of as waste in the engineering system, systems that don't actually have a function. And that's true also that the systems must be multifunctional. So spiders are really, really cool. If you've ever been a young child and pulled a leg off of a spider, you'll notice that typically when you do that, the spider goes. And that's because the spider uses a really involved system of valves and pressure gauges to control fluid flow through its limbs. And it actually uses this hydraulic system to extend its legs. Am I getting feedback on the mic, or is that still good for everybody in the back? Okay, good. So it doesn't actually have extensor muscles. It only has muscles that can flex its joints. And whenever it wants to extend its joint, it actually has to pump fluid into that limb in order to extend it, which is a little odd, particularly when you think that the hydraulic system is also the circulatory system for this animal, right? So it has a second function. It has to pump oxygen around. It has to provide fuel and hormone delivery as well. And so really, if you were going to say, well, what is this hydraulic system optimized for? It's probably not optimized for any one particular thing because it's very unlikely that the same things that are necessarily good for this are going to be good for sort of removing and supplying fluid when you need to move your legs. And then also animals must follow an inherited plan, so you get really odd, wacky things like what's this in the back of a whale? Well, this is the hip bone, right? And even though they don't have legs anymore, it's still hanging out there. And this gets even more elaborate when you look at comparative vertebrate morphology. Here's a whole bunch of limbs from animals doing different kinds of locomotion, flying, digging, running, grasping. And if you notice, they're color-coded so that all the parts of the different limbs have the same, are the same in each different animal. And you can see, so all these different animals are basically using the same constituent building blocks, changing their shape a little bit, putting them together, changing their angles maybe a little bit, but they still have the same tools. They're still tinkering with the same set of constructs in order to get these different functions. And so that makes you wonder, really, these probably can't be optimal for all the modes of locomotion. And so there's this constraint coming from a shared history. So this really leads to the idea that if you just sort of blindly copy nature, you may not actually be getting the function that you want. And that's because natural selection is not engineering. Natural selection is acting on the variation that is present in the world, and evolution acts on a principle of just good enough. What it really is concerned about is fitness, and performance is not necessarily fitness. So I grew up in Montana. We know this very well. If I'm being chased by a bear, I don't actually need to run faster than the bear. All I need to do is I need to make sure that I can run faster than my friend. Because if I run faster than my friend, my fitness is preserved. So this is the idea that evolution, frequently you can't find a system where in a biological system where you're just getting optimality for a specific singular function. Good hypothesis, though. So what can we do? We've learned that we need to be a little careful with how we look at biology and do engineering and then try to get at the science underneath that. But really, there's this idea then we need to go about this in some systematic way. And so that's what I want to get at today, because really as bio-inspired technologies, particularly robotics, has developed, we've really been able to sort of formalize this a little bit more and actually gotten to the point where we're taking a very systematic approach to looking at how to do bio-inspired design and bio-inspired science, which is actually why I'm in a physics department here. So to touch on this today, I wanted to go in and look specifically at robotics because it's what I know the most about, because I study locomotion and the science of movement. And so in 21st century robotics, there's sort of three big challenges. So this idea that we want robots that can do really good reconnaissance, we want robots that can do really good search and rescue, and really good exploration to do things like go to Mars. And so in order to do that, I'm going to touch on what we call enabling behavior. So 4 things that we might want robots to be able to do in order to not just sort of stand there. One of those is to move around, not just quickly, but stably. One of them is to maneuver, actually get somewhere where it wants to be rather than where it doesn't want to be. And then the last two sort of deal with getting off the level surface, adhering and climbing up things, and flight to actually get up into the air, particularly when small. And what we're going to talk about is how biology, so this is sort of the view from maybe about 8 to 10 years ago. I started developing this talk around that time with the guy that I was working with at that time, Bob Full at UC Berkeley. So I should acknowledge him here and say that he's been really instrumental in working out these ideas. And a lot of these original ideas came from work that he did back in the 90s. And they're coupling this idea of biology and engineering as sort of this reciprocal relationship, which is that when you look at biological systems, you use the principles and analogies from biology when advantageous, but you integrate those with the best human engineering. If titanium is strong, use it, because that's not present in any biological system. But then the converse is true, and this is where the science starts to get exciting, which is this idea that we can use the engineered systems, and we can use the approaches to understanding engineering to generate novel hypotheses and allow novel measurements of the biological systems. And this is great, but it doesn't get at one important thing, which is how. How do we do this? How do we use principles? When do we know that they're important? What are the mechanisms of the biological system that we want to adopt to the engineering system? And when we are starting to play around with parameters in the engineering system, what is that maybe telling us about the function of the biological system? And that's because what we're really looking at here is a mechanistic science. And so instead of just having this coupling between the biological system and the robotic system, the enabling behavior we're interested in, we really want to try to extract from both a mechanistic principle. And this is the idea where physics of living systems comes in. The idea that not only can we look at a biological system and we can use the principles of what we call neuromechanics, the brain and the body, in order to inspire new robot designs, we can actually use the robot to do experiments, and we can explore novel conditions, and we can use these actually as physical models and as test beds for new science. But from both, what we're extracting out is these new principles of movement and dynamics that enable behavior in living and engineered systems. And so what I'm going to do now is talk about 4 examples of those. And that'll take us through the rest of the talk. And in each case, we're sort of doing a slightly different thing. In the first case, we're trying to expose emergent principles. What that means is we're trying to take complex systems, which is most any natural system, though not necessarily complicated systems, and we're trying to say from that complexity, how do we actually sort of get emergent simplicity? That is, how do we get how do we understand how these various complex systems might all be behaving similarly? Second, we can investigate how living systems interact with their environment. So animals interact with almost all the environments on this planet. And the interaction can lead to really interesting insights into how they're maneuvering. Finally, we can discover versatile materials. There's a lot of interest in this word right here in engineering. There's whole departments on this. And one of the ways that I like to explore this is actually by using evolution, by using the diversity that's out there to inform what might be the best design principles for materials. And finally, we'll identify key innovations and extremes in function, and that'll get us back to flight. So the first thing I want to talk about is stability. And this comes from, this is a video that inspired a lot of my dissertation, actually. This is a cockroach, and there's a little cylindrical tube with a bit of gunpowder in it. And this is slowed down about 25-fold, and that cannon on the back of that cockroach has just gone off. And that seems like a little bit of an odd experiment, right? Why would you do that? And in fact, Dave Barry, if anyone remembers him, he's a columnist. He was like, how do you know when science has gone too far? You know when science has gone too far because they start putting cannons on the backs of cockroaches. But it tells us something interesting. And if we look at the recovery of that cockroach from that perturbation, it turns out that the cockroach begins to stabilize in less than 10 milliseconds, which is faster than its brain could have detected that it's been perturbed and told its body to do anything. All right? So this isn't even just a really rapid knee tap reflex. You know, we have all these reflexes where you like tap your knee, sends a signal to your spinal cord, that signal comes back out and changes the activity in a muscle. This all happens faster than that. So what's going on? Well, the idea was, well, this system is moving. It's a dynamical system. So maybe it has some stability properties that are inherent to it in and of itself. And so we decided to look at that. And you can go back and you can look at how animals run, and you find some very interesting conserved ideas, which is that almost any animal when it's running uses alternating sets of legs. So we alternate our right and then our left leg. Dogs alternate the front and back legs in pairs of 2. Cockroaches use an alternating tripod, so front and back on one side, middle on the other side. Crabs do the same thing with alternating sets of 4. And you can sum up the forces on all of these legs, and you can represent it as an upside-down pendulum, an inverted pendulum that's bouncing up and down on a spring. So this is called the spring-loaded inverted pendulum model for running, which is a bit of a mouthful, but it's pretty simple. You're bouncing like a kangaroo. And the interesting thing is that that means that stiffness is going to be really important, right? And of course, a larger animal is going to be stiffer than a really tiny little animal, but we can non-dimensionalize that. And this is a way of taking a value like stiffness that has units and making it unitless such that it could scale across all different systems. And when we do that, what we can look at is the effective stiffness that all different animals are running with. And what you'll see is that everything from a cockroach to a kangaroo, and actually all the way up to probably a T-Rex and even a human, are all running with effectively the same stiffness as they're moving along, the same non-dimensionalized stiffness. So they're all sort of converging on the same dynamics, even with the same effective stiffness that they're moving along. And we can do the same thing in the lateral plane. So we can look at these biological invariants, and we can say, well, okay, if the animal's bouncing up and down, what's it doing in the sideways plane where it might actually be going unstable? And what we can say is, well, we can take those 3 limbs of the cockroach, we can add up the forces, and it looks like a spring mass system that's bouncing into a wall. And then when one tripod picks up and the other tripod touches down, it's like it's bouncing into the other side. And the only sort of thing that's a little more complicated here is that the center of pressure, where the some forces are acting, is moving in front of and behind the center of mass. But what I want to point out with this system, particularly for the physicists in the audience, is that there's no damping, right? This is a totally energy-conserving system. In fact, it also has holonomic constraints everywhere. So you can describe, there's nowhere for, the idea is that there's no real place for energy to go if you put energy into the system. So if you perturb it, you start wiggling it in some way, where's that energy going to go? In fact, there's a theorem that says if you just have energy conserving and holonomic constraints, you can't be globally stable because there's nowhere for that perturbation energy to go. So let's simulate it. Or actually, these are my colleagues, Phil Holmes at Princeton and his group, simulating this spring mass system. And so again, it's a very simple system. We're just going to define these parameters. We'll actually fix them to the value of the cockroach. And then we'll run this simulation. And so here's the simulation of the system moving along. Here it's going to turn red when it's poked, and you'll see it'll start to oscillate. And it changes its heading. It's neutrally stable in heading, meaning it can go to any heading, but it doesn't just start to oscillate wildly. But the rotational velocity damps down and is asymptotically stable. And so in fact, this system is globally stable. So did cockroaches just disprove a fundamental theorem of physics? Not really, but it did lead to new ideas of how dynamical systems may work. And that's the idea is that there's one tricky thing about how this system is conserving its energy and where the constraints are on this system, right? There's a critical point here, right here, where it transitions from bouncing to the left to bouncing to the right. So that nonlinear transition, you can think of it as if you write the constraint equations on where this limb is acting, they're changing sign at this critical transition. And this is what's called a hybrid dynamical system. It's energy conserving, but only in a piecewise sense. And it has the same set of constraints, but only in a piecewise sense. So during this phase, it has the same constraints. During this phase, it has the same constraints. And this single transition here is actually sufficient to introduce global stability. And this has been a whole new, sort of the idea of hybrid dynamical systems, when they're stable, when they're not stable, and how you control them, has been one of the really rich areas in research of dynamics for the past couple of decades. And it was certainly not entirely because of looking at the cockroach, but this was one of the first model systems where we really saw that this hybrid dynamical effect could be really important for the actual locomotion in the real world. All right, so, well, if that's the case, we're not done yet, right? We just said that it could work. We did a little mathematical dynamics model. So let's build a robot. Or actually, our colleagues at Stanford built a robot. So this is Sprawletum. It has no sensing of the environment. It's just going to oscillate its legs with little pistons as alternating tripods. And it's going to instantiate those dynamics that we talked about. And here it is running along. And so you can bang it, you can whop it. And again, it's not sensing that it's being tapped or anything. It's just relying on the dynamics to stabilize. And so that's great. We sort of have a physical model that's sort of showing that we can use these dynamics. But that still hasn't told us if the cockroach does it. And this is where we start to get back to that idea that we're not just doing engineering. We're not just trying to build a system. And not to say that the engineering is bad. This is a really cool robot. It was one of the ones that sort of drove a lot of legged locomotion. It's about 12 years old now. Well, even longer, 14 years old now. Almost 15. So. But we want to bring it back to a hypothesis about how animals may move. And so in order to do that, what we want to say is, well, there's this new hypothesis. We've said that cockroaches use these dynamics. They may be running stably. So this was actually one of the projects in my dissertation, was to run cockroaches over rough terrain. You know, this is one of those things you go to graduate school, you never know necessarily what you're going to do. You're going to run cockroaches over rough terrain. Okay. All right, so here's the slowed down version of the cockroach running over rough terrain. And what we're going to do is we're going to say, well, if it really is relying on those dynamics in order to be stable, that means it's just driving its legs in that alternating tripod. That means the brain shouldn't be changing the commands driving those muscles in any way. So if that hypothesis of passive self-stabilization is true, we should be able to tap into the nervous system of the animal while it's running along, record the activity that the brain is sending to the muscles, and see if it's changing when it's running over flat surfaces versus running over this rough surface. So what we do is we go, so we're going to measure the commands from the brain. And what we're going to do is we're going to go into the muscle here, and we're going to record from one of the muscles in the leg that we know is involved with going up over obstacles. And we're going to look at how it changes. And I'm going to show you how we do that with a volunteer. Who wants to be a cockroach? Oh, good. Somebody was very eager. Come on up. All right. Let's turn. So it turns out brains are electrically active. You've probably heard that. And neurons are electrically active, so the signals coming from your brains are electrical signals. And muscles themselves are electrically active. And I actually like to think of muscles as neurons that do stuff. That is, they actually take those electrical signals and they transform them into mechanical work. And so if we record electrically from a set of muscles, we should be able to see how they change. So you want to roll up your sleeve for me? And give yourself a really good rub right there. Get off all the dead skin. All right, and on the back of your hand too. All right. So these are little electrodes. If anyone was here a couple of weeks ago when Flavio Fenton gave his talk, he stimulated the arm, right? So if you impose an electrical stimulation, you can get a contraction. We're not going to do that. We're going to record the actual signals that are coming from your brain to your muscle and look at the natural electrical signals in the system. Cool. So here's one. Here's two. And where I've put these two electrodes is on the muscle that controls the clenching of the fist and the rolling of the wrist. So if you flex your wrist forward, you should be able to feel your muscle here contract. And then we're going to put an indifferent electrode on the back of the hand. Okay, come on over. And what we're going to use to record are these spiker boxes, which was actually built by a set of physiologists who became engineers. This is a commercial product that you can buy now if you want to play with neuroscience at a home. They have a whole bunch of experiments you can do with kids as young as like second graders. It's a really good system. Let's step forward a little. And what this system is going to do, we're going to record the electrical activity, and we're just going to take that voltage. When you guys come down afterwards, I can show it to you actually on the iPhone app. That's why I have my phone up here. But right now, we're just going to listen to it. And we're going to see if this can get loud enough. Hold it up to the microphone, maybe? All right, so now what I want you to do is put your arm out like this. And I want you to contract your arm back like that. All right, so we're listening to the activity from his brain activating the muscle in his arm. And I really encourage you guys to come down afterwards so you can see these traces. They look really interesting, but they're fairly noisy. If you hear this, it makes a really nice, it makes a sort of a bit of a staticky sound. And that's because there's a lot of different neural signals. You got a microphone. I'll be quiet. There you go. Nice. All right, great. And one of the things you can do with this is you can EMG arm wrestle. And so if you're interested in doing that, come up afterwards. We'll set up 2 competing spiker boxes. And the nice thing is, and here's the test question, women have the advantage. Why? All right. All right, thanks a lot.
Thank you.
You can keep those on if you want to try and do more afterwards, or you
Okay.
can take them off. We can do other muscles too. All right, so let me turn the volume down. So we did the same thing for the cockroach. We recorded the electrical activity. And one of the nice things about working with insects is I told you that they're a bit less complex. They're not necessarily less complicated. They have a huge behavioral repertoire, but they have many fewer neurons innervating their muscles. And so when we look at their neural recordings, you wouldn't hear what we just heard. What was your name? Andrew's muscle. We would hear something that would sound like pop, pop, pop. And that's because the muscles in a cockroach leg are activated by single individual action potentials. So these digital-like events that are all or none spikes of activation. And they always get these spikes of activation. And during running over flat terrain, you'll see that generally this muscle has 2 or 3 spikes of activation. Each burst here is a single stride. And when we do it on the rough terrain, we see that there's a similar number of spikes that's occurring. And we can compare those statistically and do a bunch of controls. And we show that there's actually no difference that we can tell between the signals that the cockroach is sending to this leg as it's running on flat terrain versus running on rough terrain. So that's great. So what that means then is if we take another robot, we should be able to somehow scale it up such that it can run over a rough terrain as well. So this is the robot Rex. This has become one of the major experimental robotics platforms now for legged locomotion. And when it was first developed, it was the fastest legged robot for its size, scaled to body size. And you can see it's running along. And the really interesting thing about this is it has no sensing of the environment. It's not detecting anything about the environment. It's like that simulation. It's sort of going in random directions as it bounces along. You can put a controller on it to direct it, but that's a different story. But it does have this spring leg design, and if you slowed it down, you'd see it's running with an alternating tripod gait. And in fact, what the engineers first did is they said, I'm going to do better than the biologists. I'm going to do better than the physicists. I'm going to optimize. I'm going to optimize the parameters in this system for the fastest speed I can get. That's what they got. They tuned the stiffness. They converged on the same value that the biological systems had already been at. So this was good. We are actually, the engineers, we were all very happy about that because it was nice that the system could get to that point. So then we could take that same robot, and I do that very frequently, and we can run it now over the rough terrain. And so this is sort of a sufficiency test, seeing if that passive dynamical system that just has the alternating tripod gait could run across a scaled up version of the same terrain. So this is the exact same terrain, just blown up to Rex size. All right, there we go. And just to show that it doesn't have to be hexapedal motion, you can sort of do similar things with Rex running in different gaits. Oops. My clickeroos. There we go. So by changing the controller, you can get it to be bipedal. All right, so what did we get out of all that? I mean, that was maybe a little entertaining, but what we wanted to point out is this idea that we had a biological system and enabling behavior that we wanted to generate in the robot, and a scientific principle we wanted to get out of the system. And so we were looking at stability, and we made the observation that cockroaches seem to be able to run over rough terrain very stably and potentially without with modulating their motor commands. And what really came out of this was this idea of hybrid dynamical systems really instantiated into locomotor systems, that you can have these stability inherent in the dynamics of how animals run, even without any damping. So I'm actually going to skip this section because we're moving on a little bit. If people want to learn about how cockroaches navigate their environment, we're using their antennae as tactile probes. You can come talk to me a little bit later. But it turns out very simple control laws can tell you how the animal's brain has to be processing information as it's running through the world. And I'm going to skip forward, and I'm going to talk now about sticking and adhesion. So one of the real interests in robotics is to get off the ground. And one of the ways to do that is to adhere to things. And animals are really good at that. So here's a vertical wall, and that's a gecko. And that's in real time now. And if we slow it down, you can sort of see the body undulating as it goes. This was with a very early high-speed video camera, so it's not very good resolution. We can get a slightly better view now if we go forward. So here's the gecko running up. So this is vertical. It's not flat. It's sort of hard to tell. In fact, the kinematics are very similar. And what it's doing is it's attaching and detaching its foot pads in about 40 milliseconds, which is remarkably fast. And one of the things, one of the projects I was involved with actually when I was an undergraduate was looking at how they're able to adhere to these walls. And it turns out that if we want to do that, it's a little challenging because there's a lot of geckos. So which one do you study? And one of the ways you can answer that question is you can say, well, maybe we should study the ones that are particularly good at things. And so we can plot clinging ability versus body mass. And you can say now, well, who would you study? Who would you study? It's number 6, yes, the one with the greatest adhesion. Sure, the smallest could be interesting too if you wanted to see how miniaturized the system could get. I think that's another kind of extreme performance. But for now, let's focus on 6. And 6 is this guy. This is the tokay gecko, gecko. And this looks like a really nice expression, but if you've worked with these guys before, you'll know that if you see this expression, you are about to get bit. So it's not very pleasant. We usually, whenever somebody comes into the lab, first work with them, we usually have to hold their hand in the chamber and just let the gecko bite it around a glove for a few times just so you get used to it. It doesn't hurt. It's just very surprising because the gecko barks at you when it does that. All right, so if we take a look at this guy and we take a look at its foot pads, and we see something really interesting. The foot pads are sort of arranged into these arrays of lamellae, so ridges of hair-like structures. And we can zoom in on those hairs, and what we see is they come in clusters. So there's about 1 million foot hairs, or setae, per toe. And on each of these individual setae, they split. So each seta is about the size of your hair. And then it splits 1,000 times down into these little spatulae. So they have billions of spatulae. And it turns out that these spatulae are only 300 nanometers wide. That's smaller than the wavelength of visible light. And because of that, it seems like the gecko can create such intimate contact with the surface that it's able to adhere by van der Waals forces. They would fall off very quickly as you got further away from the surface. And so we did a bunch of tests to sort of show that van der Waals are sufficient to explain all of the adhesive ability of these guys. So that's really interesting because van der Waals are particularly interesting particularly interesting way to adhere because they act between any two surfaces, pretty much. As long as the system is polarizable at the quantum level, you can get van der Waals forces. So basically anything that's not Teflon, and geckos even sort of stick to Teflon. So we wanted to look. So we want to say, well, okay, there's a lot of interest then in building an adhesive that can work like this because it's dry. It adheres under almost any condition. It can adhere in vacuum. It can adhere in space. It's reversible. It ends up at self-cleaning. If you put dirt on it, the dirt preferentially sticks to the surface rather than the adhesive. It also gets stickier as it slides, which was work that was done with one of my colleagues who was a graduate student here. So Nick Ravish. So we discovered that as the gecko feet slide, they actually get stickier, which is sort of the opposite of what you would expect from normal friction. Kinetic friction is lower than static friction, right? Not in this case. And so there's a lot of interest in generating these artificial synthetic gecko adhesives. But how do you do that? What's important? How do we actually, should we just copy this seta? Well, sure, we could do that, but again, there's a lot of different geckos, which means there's a lot of different toes. Not only are there a lot of different toes with a lot of different morphology, there's a lot of different setae. And they have a lot of different things to them. They pack in different patterns. So sometimes they're really dense, sometimes they're big, sometimes they're small and really compact, sometimes they're very curved, sometimes they're very long, sometimes they split into lots of little spatulae, sometimes they don't. So it's sort of like a Dr. Seuss story, right? Which one do you study? And the idea then is, well, maybe we can find some principle that addresses all of these systems. So this was done, and I'm going to bring up this paper by Arts in 2003, a really interesting paper because it said, let's collect all different kinds of animals, not just lizards, that adhere with hair-like structures. And let's plot them on a curve. And what we see is that as body mass increases, so as presumably the amount of adhesion they need to produce goes up, their spatular density goes up. That means they get more and more of those little tiny hairs in there. So maybe what's critically important is that we make it, if we want the stickiest adhesive, we make the one that's the densest. All right, we tried it. Or actually, our engineering colleagues at Berkeley tried it. Again, I'm not an engineer. And there's a problem. The hairs stick to each other as they get really, really dense. Yeah, good idea. All right, but wait, why? You know, what's up with this principle? And one of the things is that there's something very particular about these points that I put up here, and that they're different species. So I put the density of different species up there and the mass of different species. And you have to remember, biological species, again, have evolved. And they've evolved from shared common ancestors, which means that these points are not independent. So you can think about to get at this, if I just take 10 species, so these individual points are different for different species, and I just take 2 different values, let's say body mass and spatial density or something, and we plot them against each other, we can find a nice regression line, right? Well, what if I told you that all the blue guys are related to one another, and all the green guys are related to one another? And what if I told you that the phylogenetic relationship looked like this? All right? Well, now if we go into the blue ones and we do a regression, we find that there's no relationship. And if we go into the green ones, we find that there's, we find that there's no relationship. And so this is the idea that species are not independent. They share a common history. And we can do that here. So my colleagues, Ann Peaty and Bob Full, did this a really wonderful paper that I wanted to highlight here, which is going back to this master curve and filling it in a little bit. So the first thing they did is they collected a lot more beetles from the literature. Lo and behold, here's all the beetles in red. Not much of a relationship. Now let's collect a lot of spiders, because there's a huge range of size in spiders going all the way up to things that are close to the size of that ambler pigeon that I talked about earlier. Here's all the spiders. All right, not much of a relationship there. And in fact, if you look inside flies here, this is really driven by this measurement out here. It turns out if you remeasure that, there's various reasons why that one was wrong. It's debatable, but even if you ignore it entirely, the rest of the relationship, there's nothing there. And we can collect dozens of other lizards and show that within each lizard family, there's not a relationship either. And so what's really driving the differences here? Well, there are differences between groups of animals. And it turns out that all these guys up here are dry, and all the guys down here are wet adhesive. So they're adhering by different mechanisms. And in fact, it probably makes sense that if you make a really wet adhesive, you can't get super dense or you'll start to stick to each other. And so that suggested that maybe there's something else that's important about what's going on. And again, you go back to this idea that it's van der Waals forces. One of the things with van der Waals forces is it's not very chemically dependent. As long as you get 2 surfaces pretty close to each other, they're going to adhere by van der Waals forces. But that's the critical bit, is you have to get them close to each other. And so what's really important is the geometry. And so if you look at, so this is a single gecko toe hair. And what happens is if you just push it into a surface and pull it away, you won't get any adhesion. But if you push it into a surface and slide it a little bit, and then you pull, now you'll see this little cantilever beam will get pulled away. And so this preloading, which allows the sort of tips to conform to the surface and make intimate contact, is critical for inducing adhesion. And it also ends up being, so you can see the cantilever going well past where it was originally. So it's producing active adhesion in this direction. And it pops off at about 20 micronewtons for an individual hair, which means that a gecko, if it was adhering to its theoretical maximum, I could take 2 geckos, 1 in each hand, and it could support my weight on the wall. So I won't tell you how much I weigh. You can figure that out yourself. All right. It turns out that geometry is important for detaching, too. And there's a really interesting property that regardless of how much they're adhering, so even if the hair is preloaded or not preloaded, you can change the amount of adhesive force. As you pull at the hair, once this angle between the hair and the surface reaches 30 degrees, the hair pops off. And sort of creates a peeling mechanism. So maybe what's really important is not necessarily all the structure of the gecko adhesive itself, but how the gecko deploys its adhesive. So let's look at how the gecko deploys its adhesive. So here it's moving along, and take a close look at those feet. They're doing something a little odd. We can zoom in a little bit. Here's a zoom in through a glass slide of what a gecko toe is doing. It's rolling up like a party favor. So it's actually peeling back from the front. And then when they press down, and I don't have this video here, when they press down, they do the opposite. They deploy out, push the toe pad into the surface, and actually slide their toes in a little bit. It engages all those hairs. So maybe we don't need something that's particularly dense if we want to build a good robot. Maybe we just need something with decent geometry that has this induced adhesion with preloading. So our colleagues at Stanford now make these tips that have 45 degrees angles. These are quite big. They're about 3 orders of magnitude larger than the gecko hairs. And then you put them on a robot. And so this is StickyBot. This is one of the robots that was developed out of this collaboration by Sung-Bae Kim and Mark Kukowski. And you can see that what it's doing is it has that gecko-inspired adhesive on it, and it's deploying it in sort of a similar manner in order to get that preloading. And a sticky bot has a number of other innovations in it that are very cool engineering, but aren't particularly relevant to today's talk, which is about the biological inspiration part. So you're able, if you're sort of looking at these, again, very complex hierarchical biological materials and systems and trying to understand what's actually important about them for extracting out the particular aspect of performance that we're interested in as scientists. And so in this case, again, we had this observation that geckos stick to myriad surfaces, and we're really interested in adhesion, but there's this idea that sticking via van der Waals really requires smart geometry and proper foot loading and unloading. And really, we can do a lot with different materials to adhere that way, as long as we get that part of it correct. Now, I should say that there's a lot more that's been done on this. There's been literally thousands of papers written on designing new gecko-inspired adhesives. So there are a lot of ways to tune that and make it specialized for different systems. So there's still a lot of work to be done. The last thing I want to talk about, before I let you guys come up and play with the demos, and I'll go through this in just a couple of minutes, is flight. Because flight is really exciting, and it's one of the things I'm spending most of my time now studying. And flight is easy if you're a plane, sort of. Flight is maybe possible if you're a bird. You can still sort of understand it with good old Bernoulli and some flapping wing ideas. But you've probably heard that this idea that honeybees can't fly. Maybe somebody's told you that before, that according to classic aerodynamic theory, whatever that is, honeybees can't fly. And that's not true. If you solve the Navier-Stokes equations in a computational fluid dynamics simulation, they will fly, which is good for the honeybee. But it is true that you can't, what they really are saying is that you can't use steady aerodynamics. That is, you can't just sort of average over an entire wing stroke and predict what the forces are that should act on the wing. And if you do that, you'll get a model that won't predict sufficient lift for a honeybee to take off. What's really important is how the wing and the airflow varies through time, so the unsteady aerodynamic effects. And so there's been a lot of interest over the past 20 years now about how insects fly and if we can build a small robot that might be able to mimic this kind of flight. And this is incredibly challenging, not just because it's very hard to generate sufficient lift to do so, but flight is inherently dynamically unstable. There's two unstable modes, regardless of whether you're a plane or a flapping insect, at least two. One is this phugoid motion where you pitch forward and you accelerate, and you sort of get these destructive pitches and oscillations forward. If you've ever piloted a small plane, you may have felt this early on in your training. It's not good. The other one is a slower mode that is unstable laterally with a roll, so it sort of falls this way. And so these animals fly along with their wings, and the first thing I want you to notice is how their wings are actuated, because it's actually remarkable. It's different than you would expect, and it's not actually that complicated. They have these large power muscles, and the power muscles don't attach to the wings. Instead, they attach to the exoskeleton, and they deform the exoskeleton like an elastic shell. And so then the exoskeleton so this is like just the center part of the animal, its thorax, and as this muscle contracts, it'll pull in the front and the back of the shell, and that will cause a pattern of strain that causes the wings to go down. And then these up and down muscles will contract, and they'll cause the upstroke. This is a very simplified version of the joint, and where you do get a lot of complexity is in how this joint is structured. Because in an animal like the hawk moth here, during each cycle of activation, the brain is only sending a single pulse of activation to these muscles. It's only going bang, bang. It's creating the downstroke and the upstroke, all right? Where you're getting these complicated wing motions that you see when this animal is flying is through that wing hinge. And so that single bang, bang, bang of the muscles is producing this complicated twisting, rotation, and translation. And so if you were to trace out the wingtip here, you would see that it's moving in a figure-8 pattern, where as it comes forward, it flips back, and then it translates backwards and it flips forward. And note the plane here. This is, gravity is this way, right? So the plane of motion is not how you would think you would fly, which is like getting in a hang glider and flapping the wings. It's actually upright like this, flapping the wings back and forth like this. So the vortices are being shed downwards. All right, so people set out to study this, and maybe my favorite study of looking at how insects flew was this work by Michael Dickinson in 1999, which is to say it was really hard to image fluid flow around a fly. So let's not do it around a fly. Let's build a robot that helps us. And so this is the robot RoboFly. It doesn't look like much of a robot because it's really meant to ask, how does fluid flow around wings? And what I haven't shown you here is scale bars. This artificial wing on the robot is this big. It's much larger than a fly. In order to get similar fluid flow, you have to put it in a different fluid, because when you scale up like that, you need to be in a fluid that is much more viscous so that it feels like moving through air when you're much smaller. And so it turns out you have to submerge this whole thing in a big tank of mineral oil, which is actually convenient because now you have a fluid, a liquid around it that you can put seeds, you can put imaging particles in, and you can image that fluid flow as these plates move around in the fluid. And so you can see these plates moving along, and you can see the vortices forming as the plates move through the fluid. All right, so what did they discover? Well, they discovered two really interesting things that are what we call unsteady mechanisms for fluid flow. And one of these is that normally if you have a bluff body like a wing, an airfoil here moving through the air, you have a small suction force that's effectively pulling the wing forward. When you have a very steep angle of attack, so you're way up like this, and you're really thin, it turns out that that suction force changes direction and ends up acting in conjunction with the normal force. And so you get this force on top of this force, and you'll get more lift that way. This is called a leading edge vortex. You can think of it as there's a vortex of air that gets caught onto the leading edge of the wing and just circulates there and helps pull the wings up, pull the body up. The second thing is that when the wing flips, it's going to shed the vortex, and it's going to shed a trailing vortex as well. So what happens is as the wing goes back, a vortex forms on the top. Then as it gets through to the front of the wing stroke, that vortex is going to shed, and a sister vortex down from the tail of the wing is going to shed. And the interesting thing is because the direction of these vortices, the fluid flow is jetting between these two vortices. So you get an increase in velocity in the direction of these blue arrows. And the particularly cool thing is then on the return stroke, the wing actually re-interacts with this wake, and such that it recaptures the vorticity here, and now you have the jet here increasing the speed of the air going underneath the wing. And that increases the lift on the wing. And that's called the wake recapture effect, or the wing-wake interaction. And they were able to discover both of these effects. This one had been hypothesized before. This one had been thought of a little bit, but was really discovered in doing those experiments. And so then the idea is that maybe we can use those ideas to make robots that are flying. And if you guys saw this paper in Science from last year, this is Rob Wood at Harvard, starting off work that he began actually at Berkeley with Ron Fearing, making a small micromechanical robo-bee, they called it, but really it's more of a fly based on this drone fly. And what you can see is it's fairly simple. It's doing a translation and rotation of those wings with a very simple piezoelectric motor here. So it's just these two sort of virtual flight muscles that when you pass an electric current through them, they shorten. And they create this complex sort of translation and rotation pattern, very much like the drone fly here. And so they were able to then take this and actually get it to successfully take off, support its own weight, produce sufficient lift, and it could actually maneuver around a little bit. And in fact, the first thing when I saw this video is I was like, well, what are all these little dots on it? Those are actually motion capture markers. This whole thing is in a Vicon camera system, so they have a bunch of cameras around the system telling the robot where it is in space, and that's actually how they stabilize. So it's not fully autonomous in its sensing yet. We still have a long way to go for that. But you can see this is actually pretty remarkable. And you might say, well, the crashing is pretty funny. You know, that doesn't seem very biological. But if you ever walk into a room with a bunch of bees and you're worried about getting stung, turn out the lights. You'll hear thump, da-da-dum-da-da-da-da-dum. Because bees require vision in order to stabilize their flight. And they can't see very well in the dark. And so you can actually show that as a bee is flying along, if you turn out all the lights on it, it will pitch catastrophically, go unstable, and fall to the ground. But it's perfectly fine. That's, in fact, how they choose to land at their nest frequently is they just crash into it. Because when you're small, crashing like that doesn't tend to matter that much. All right, so the last thing I just want to say is that's great, but it's still a sort of far cry from here. Look at the maneuvering that these guys are doing. All right, so we still have a long way to go, and I think this is really exciting, is that we're still trying to understand how muscles can be controlled to modulate that wing stroke. And one of the things that we found is that you would expect these muscles from like a moth here, that if we looked at the power output from the muscle, we can change the phase of activation. We can take the muscle out and do materials testing on it. And we can say, well, that muscle, if it was really a power muscle, we would expect it to produce the maximum amount of force possible, or maximum amount of mechanical power output possible for a single spike. And it turns out it doesn't. The animal activates that muscle at only about 50% of its maximum power output. And when I saw that, I thought, oh, that's sort of interesting. What does that make you think of? Why might it be acting here rather than here? Anyone? What's different about this part of the red line than this part of the red line? Yeah, it's linear. Somebody else? Yeah, there's slope there. Good. The derivative's high. And so, well, that's great. I mean, the derivative locally is 0 here, but it would fall off a bit. But there's something particularly interesting about this point, which is that if the animal were to modulate its phase of activation around this point, it would be able to do up to 200% of its normal power output, or get its power output all the way down to 0, with only about a 4 millisecond timing change, which is remarkably precise for a motor system. And so we set out to test this, and we actually showed that the motor system in this can actually define those spikes of activation down to 1/3 of a millisecond, which is the most precise neural signal we know of, particularly in a motor system. And then it can actually modulate the timing of that activation all through this range. So it looks like these muscles, even though they're power muscles and they're only getting bang, bang control, that bang, bang control is phase modulated in order to do a lot more. And we actually showed that is sufficient to induce a turn. When you're in an animal, you can reprogram its muscles to do a turn. So I just want to end with this, this idea that it's a really exciting time. We have a lot of engineering yet to do. We really don't have engineering systems that have the capability of, say, this dragonfly to maneuver in and land on a small little perch and perch there. And we don't really understand how these guys' brains and bodies are working to process information about its environment. The reason this guy is perching here is he's watching flies go by, and he's waiting to plot his intercept course such that he can fly and meet that fly. But of course, there's a lot of other behaviors that animals have as well. And so we really need to understand how maneuverable these guys are in order to be able to get away from another predator. And so you may have seen this video. This was actually my colleague in the lab that I worked at the University of Washington. I filmed this. We were out and set up our high-speed video cameras at a pond to film a bunch of insects. And he heard a splash, and he hit the trigger. And he wasn't even looking at his camera, and he heard it hit a trigger. And that happens to be what was recorded on the camera when he hit that trigger. And this is a really interesting thing. Look at him. Even though he doesn't get it, his hands come forward, right? He had another example of that sort of feed-forward kind of behavior that animals do. All right, so we learned a lot about how flies can fly and how unsteady aerodynamics can be important about that. And I just want to end by saying, look, principles and analogies from biology are great for inspiring engineering. Be a little careful with it, though. Only use them when they're advantageous. Engineered systems are really, really good. And this idea that we can then use robots, I would actually argue that I will do better science if I'm working with a roboticist than if I don't. So we'll do better science if we actually use experimental robots to ask new types of questions about living systems. And we can even reveal new physics about how complex systems work. And finally, as nature-inspired technologies emerge, the scientific process itself becomes a strategy for innovation. And that's great, but that really means that biodiversity is even more important than we thought. It's not just save the animals now, it's save the robots as well. Because if we lose the innovations that are out there that we haven't discovered yet, we may not be able to create the next great robot. And we're really just getting started. It's not just robots, it's materials. I just talked about robots because I study movement. Materials are being biologically inspired and nature inspired all the time. We're looking at communications strategies that are based off of the brain, new medical innovations. It's really an exciting time to be doing that. And that's how I'll end. Thank you, guys. Come arm wrestle. Yeah.
Any questions?
Yeah.
I was wondering, in the case of the geckos, can you swap the roles of the material and create a wall that anybody could climb?
That's an interesting question. Probably, there may be some capability to do that. Part of the problem is that you would have to make the wall such that it could conform to a very rough surface, because if you looked at my hand, I have pretty smooth skin. You can come and feel it. But it's not actually really smooth if you look at it under a microscope, right? And so it would have to be structured in such a way that it could actually interface with that. And I also have like a lot of oils and such in my hand. It could be, now the van der Waals forces could still work in that situation, but it might be a very expensive wall. So interesting idea, though. It would be fun to test.
Thank you for your great presentation. I have a question about the experiment that you just did with Andrew.
Okay.
So the noise, you attributed the noise to the signal from the brain. But I think that signal from the brain is very small as compared to all of those moving parts and mechanical movement and things. So do you think most of that noise is coming from the mechanical motions there?
So there is an idea, that's an excellent question. It's something we worry about in recordings. I can tell you that no, that's not the case. It's a very standard technique that's been done for decades, and it's been really well controlled for that kind of thing. And you can actually go and you can record intracellularly across the membrane of the cell, and you can directly correlate it to the patterns that you're seeing outside. So very careful controls have been done there. The electrical signals are actually not that small. They're on the order of a millivolt or so. And we usually use these differential recordings with a common mode rejection. But particularly, one of the problems for doing it in humans on the surface is that our skin slides across the top of our muscle, and that will introduce noise. You can also, you can separate it by frequency as well. The noise introduced by motion is much lower frequency than the spikes that you're getting out of the nervous and the muscular system.
More questions for our speaker.
Yep.
The hinge that you were talking about for the flight, is that a static hinge or is it dynamic?
Yeah, okay, I see what you mean. Yes, it has other muscles. And it has these little tiny muscles that have been, you had power muscles, right? These are called steering muscles. And they're little tiny little pissy things that can't produce much force, but they do attach directly to the wing hinge. And they tilt and they twist and they trim the wing hinge very much like you would with the lines on a sailboat. And so that's how animals, these insects were thought to turn. And those certainly contribute to turning, zero question about it. But it was thought that those sort of did all the control by changing the stroke angle, for example, changing the amplitude a little bit, and that the power muscles just sort of were these dumb, big bang, bang, bang motors. And what we're seeing is that there's an interesting interaction between the two now. And one of the things that we're doing in the lab right now as we speak is we're going in and we're recording from all of those muscles simultaneously. So we put 24 wires into a moth as it's flying around in a virtual reality arena, and look at how it's processing information and changing its motor output.
Questions?
What is the van der Waals?
Van der Waals forces are really interesting forces that come, they're quantum mechanical effects that you see at the level of biology. So what happens is when you have a electron cloud around a molecule. So it's not just an electron that's going around a molecule. It has some distribution, you can think of it. And that cloud sort of has a distribution of charge. And that charge is oscillating due to the quantum mechanical nature of it. So so if you bring in two molecules now, one with sort of a stretchy electron cloud here, one with a stretchy electron cloud here that's oscillating a little bit. Now let's say, okay, freeze frame. Right. At that moment, there might be a little bit more of electron density on the right side of this molecule and a little less on this side. And that's going to produce an instantaneous time-varying electrical field. But at that frozen moment in time, you'll have an electrical field that's generated by that. And that will, when you bring something else that's stretchy and charged into contact with that, it's going to stretch. And if you get those two things close enough, those instantaneous fluctuations will basically stabilize, and you'll get binding because they'll induce a polarization in each of the two molecules. And so what we call about it is this idea of polarizability. It's how stretchy the electron cloud around molecules are. And all electron clouds are going to be somewhat stretchy. But things like Teflon, which has four fluorine atoms around it, so that the electrons are really bound tightly inside there, isn't very stretchy. And things like big complicated proteins like the beta carotene on the surface of the gecko is quite stretchy. So it's different from having a standard dipole, which is like if you have water, you have oxygen here, hydrogen, right? So you have a bit more positive charge, a bit more negative charge on one side. And so that has an inherent dipole to it. You actually show that you don't actually need to have that inherent dipole in order for these guys to stick. You can actually induce that polarizability, and that's sufficient. The trouble with that, it sounds great, right? It sounds like a magical way to adhere. The trouble with it is it falls off. So like Coulomb interactions fall off as you go over like R, and then various other forces fall off as you increase the distance squared. These things fall off as you increase the distance to the 6th power. So as soon as you get a little bit away, it just goes diving away. So you gotta get really, really close.